I am pleased to introduce Aaron Alt from Purdue Oats, who is the senior research engineer and a rancher himself. And Aaron's going to talk to you a little bit about what the prep work that's been done in the lead up to the event. So lots and lots of stuff, including up to last night. So it'll be great to hear from Aaron on where we're all at. So thanks so much, Aaron. Hand it over to you.
Thanks, Martha. I think where I live, kind of like we call it pop and not soda. I don't think I get to say I'm a rancher. I got to say I'm a farmer, but one or the other. But I do have cows, and I have corn, soybeans, and wheat. So thanks, Martha. So as Martha mentioned, I'm a co-founder of the Open Ag Technology and Systems Center, the Oats Center at Purdue University. Also a full-time farmer. So I'm a software developer and a farmer. So that's me, but we've been doing a lot of work. I feel like we've had a hackathon for about a week and a half leading up to the hackathon that's going on in parallel with this event. The goal for this whole event is fixing the soil health tech stack. I think for developers, that largely means making it less painful to interact with this data, right? So in other words, I got a spreadsheet or a PDF or something of soil sample data from one lab, and now I can get it into my chart with as minimal interaction as I need in between. And so that's kind of what on the technical side we've been working on. The stuff that we did ahead of time getting ready for this was, one, just trying to get as many examples of lab data as we could. There is a standardized format out there that the Ag Gateway actually maintains, and so thanks to them for participating in this event also, and working with us leading up to the event to help us understand what was there and what was in the standard. It's called MODIS. So that's the word to remember, MODIS. I don't think it even means anything else. So it's just this. MODIS is our standard way of getting soils data. Some labs can make it, some labs can't. And our first task leading up to this was make a tool that if you gave me a CSV file or a spreadsheet or an original kind of MODIS XML form, I could get it into what we coders would call JSON. So we can get it into something that's easy to navigate, share between tools, stuff like that. And so we did that. It's been published and released open source for anybody to use. And we took 4 different data sets actually that we have, and we've run them all through that tool so that they've all turned from the form they started into standardized form. And I'm not going to steal Chris Covey's thunder because they did all the work taking the samples, but there are some new soil data samples because of this event that Chris's group did. And so they're posted up online right now. And actually, if you want, I can just share real quick and show you where they're at. So I'm just going to share my whole desktop. You guys see it? Okay, and so this site right here is the hackathon site. If you want to come back to here and see it later, it's https://github.com/oats-center/2022 fixing soil-hackathon. If you go there, you will see this page. And at this page, we've got listed a bunch of stuff for the hackathon folks, but I'm going to scroll down to our example data sets. So here are some of the example data sets we have. If you click these links, there's a little bit of description here. But for example, the data that was created for this particular event, if you click this link, you'll see the source data here in Excel file. So you can actually open up that Excel file, see the results that came back from the lab. And then we have the automatically converted MODIS JSON form of all of that stuff right here in these files as well. And once you get here, if you click back to examples, then you'll be able to see a bunch of data that we've also done the same thing for the hackathon participants. So for example, this NYR East 50 from A&L Labs, that's one of my fields actually on my farm, a soil sample I had. And thanks to Randall Warden at A&L Labs for getting me the MODIS version of that. That's what really kicked us off, was I actually had a real MODIS XML file as a result of that. So there's a PDF file in there of my sample, there's the MODIS. And then huge thanks to Tomcat Ranch and Point Blue Conservation Science for giving us this Tomcat historic folder right here. And so in here is actually a great treasure trove of historic data that they've released into the public domain for anybody to interact with in soils. And the source data was here in this Excel file, and a few actual raw samples that came from the Ward lab. And then we've run all of those through our tool to turn them into these standardized MODIS JSON forms. So at the hackathon right now, they're working on using these things and actually making something that will take that and turn it into, for example, a standard CSV or a standard Excel file. So if you had it in whatever form it came, at the end of the hackathon, fingers crossed, you will actually be able to get it into a standard form that then you can use in other tools that know the standard form. So that's some of the work that we did ahead of time in publishing those libraries and tools and stuff like that. So with that, next up, we're going to talk to Chelsea Carey from Point Blue Conservation Science, and I'll let Chelsea take it away.
Awesome. Thanks, Aaron. Yeah, so I'm just going to spend a bit of time talking about the historical data sets and set the stage for Chris to talk about the data that was collected for this event in particular. So I'm going to share my screen and okay, do y'all see my screen? Okay.
Yes.
Great. All right, so yeah, so hi everyone, my name's Chelsea Carey. It's great to be here this morning. I'm going to spend just a few minutes talking about some of the work that we've done to explore soil health at Tomcat Ranch. And it, as Aaron alluded to, it's directly relevant to the Fixing the Soil Health Tech Stack event and the hackathon in particular. So for those who don't know, and I'm sure many do, Tomcat Ranch is a living learning laboratory that's situated on the central coast of California and has the strategic goal of inspiring the transition of 5 million acres to regenerative rangeland stewardship in California. They're really leading the charge as regenerative agriculture thought leaders and focus on advancing the science behind regenerative agriculture, on influencing the way that we all eat, that society eats, in order to create demand and accelerate adoption of regenerative practices while improving the health of human communities. And then also bringing people together into gatherings so that we can help catalyze action, learn from each other, and help that transition into a regenerative agricultural food system. I work for Point Blue Conservation Science, and we're a nonprofit that's headquartered just north of Petaluma, in Petaluma, just north of San Francisco. And we have about 160 staff members who focus generally on climate smart conservation. So building an adaptation and resilience into landscapes, helping to mitigate greenhouse gas emissions where possible. And we see a lot of opportunity to help achieve conservation objectives and goals by partnering with farmers and ranchers to in the search for regenerative stewardship. And we've had the pleasure of working closely with Tomcat Ranch for 12 or so years now as their on-site science partners in service really of advancing both of our missions. So through the tenure of our partnership, we've focused really closely on trying to demonstrate the practices and strategies and benefits of regenerative stewardship and create the enabling conditions and support the tools that speed its adoption. So from a science perspective, this is really about collecting data to help better understand how we can manage these lands regeneratively, and then not stop there, but tell the story of what we're finding so that hopefully we can inform action, inspire action at a number of different scales, from a single ranch to at the state level with agencies and beyond. So we do this in a number of ways. We co-create soil health trials, so and other experiments together. So the picture in the middle bottom is me and Mark Biaggi, who's the ranch manager, and I'm there in the oversized raincoat. But this is us working on collecting samples from a soil health trial we created together, and then just recently published some of those findings in Frontiers in Sustainable Food Systems. We also work with others to move technologies along the R&D scale. So the picture above at the top is Dan Kane, I believe Chris Covey from years ago, working through some of the methods for quick carbon at Tomcat Ranch. And then we also see a lot of value in synthesizing published literature. So that's another thing that we spend a good chunk of time doing. But what I want to focus on today is some of the observational monitoring that we've done on the ranch. And so this has really allowed us to better understand where we're at, where we're headed, adjust course through annual ranch planning with the land and livestock team as necessary. And it also, this kind of observational monitoring also offers an opportunity to assess the usefulness of indicators and really look at our theory, tap into and look at our theory of change over time. So I put up a list of indicators here that we measure to track ecological function, which, as you can see, span above and below ground, everything from birds to the vegetation to soils. And both Tomcat and Point Blue really value looking at the whole system. So we don't want to just steward for soil health, but we want to steward for soil or for biodiversity and really realize the full potential of regenerative stewardship by promoting multiple benefits. From a soils perspective, you can see the metrics that we monitor here at Tomcat Ranch, and we focus heavily on soil dynamic properties, so properties that'll change or are expected to change with management, carbon, bulk density, water infiltration. We look at a whole bunch of nutrients. I also have a background in microbial ecology, so we get to play around with some molecular assessments for microbes in addition to looking at microbial biomass and these sorts of things. And then look at some inherent properties as well, soil texture and pH. I think of as an inherent property in these places. So we collect all this information across 40 points at Tomcat, which you can see here. This is a map of the ranch, which is about 1,800 acres, mixed with open grassland, scrubland, and a couple of other habitat or ecosystem types. And in 2014, we began monitoring these points, which were randomly selected using a spatially balanced design. We follow standardized methods, which we actually developed on the ranch, and now we use across the state of California. And if you're attending the next session, I'll talk more about our effort there statewide. And this, so these methods include sampling soils to 40 centimeters for carbon, looking at 0 to 10 and 10 to 40 centimeters, and then all of the nutrients and microbes as well. We also look at bulk density at the surface and also water infiltration. So the monitoring is not meant to be static or just provide a baseline. Instead, it's meant to allow us to track change over time. And so we do go back to the same points every 3 years and resample. And this can produce insights and figures such as the one that you're looking at here, which is showing the change in carbon from 2015 to 2018 to 2021. So much of these monitoring data, as Aaron mentioned, have been made available for the hackathon, and hopefully it'll help us fix the soil health tech stack. And I'm gonna then hand it over to Chris to talk about an effort that we did on the ranch in service of this particular event. So thank you.
Thanks, Chelsea. I appreciate that. That was great. Yeah, you guys really came through with us on that lab data. Part of fixing the soil health tech stack, I think, is fixing the throughput of labs, to be honest, because we definitely hit that quickly. So you totally saved us there at the end by giving us some data for that. So so that's great. So some great conversations going on in the chat. I'm not going to repeat them all here, but some really good questions. There was one in particular which was does Modus convert Excel files into JSON? And the answer to that is Modus is the format. So Modus would describe the kind of JSON that you get, and the tool we released converts Excel files in a range of formats into that JSON format. So that was the answer to that question. With that, then, if you have some questions for Chelsea or Chris or Dorn, who's coming up, please be sure to put them in the chat or put them in Slido on Event Hub. We're watching that, and I'll make sure we get those questions asked at the end. So, Chris, I believe you are up. Chris Covey from Skidmore College and the Soil Inventory Project.
Great, and I'll just check that you all have that sort of cover slide up. And then
Yes.
I'll say thanks to Aaron and Rob and Tomcat and the folks at the Farm Foundation for hosting this conversation and helping us move all of this work forward. I'm Chris Covey. I'm an assistant professor of environmental studies and sciences at Skidmore College, and I am the president and co-founder of the Soil Inventory Project. And I'll say that the Soil Inventory Project is this collaboration. My co-founding partner, Bruno Basso from Michigan State University, it kind of handles the modeling and interpolation side. I've been working on field methods, sampling design, and methods for basically, I like to say, getting geolocated dirt in a bag from as many places as we possibly can. And we are a not-for-profit organization. Sonali Lamba serves as our executive director and helps us sort of keep this work going, along with a whole host of partners. And I want to highlight in particular, I think not just for this event, but also as kind of a leading partner for us, Tomcat has been hugely supportive of this work since even, I'll say, even before we got going. So we're deeply appreciative to them and to Rob and the others who helped get this together. So I just want to quickly kind of talk about how we see the challenge in the space. And I think like a lot of the folks on this call, we believe that regenerative agriculture can work and that you can build soil carbon and that can lead to outcomes. And I think where I'm focused is trying to put some numbers on this graph and on the right. We understand that we can change practices and we can build carbon and that can have an impact on outcomes, but it's going to be really important to quantify that and to quantify at scale if we want to link practices to specific outcomes in a way that's quantifiable, not just for markets. I'm interested in markets, sure, markets pay for things, but also for producers, land managers, folks who are active on the ground, understanding what does it cost me to do something and what do I get for it? And that get is multidimensional, and we need the data to track that. So I really appreciated the question in the chat about linking all these other pieces of data, hugely important and something that we need to work on as well. Just how are we getting data? We've got a bunch of ways that we're trying to bring data forward in the Soil Inventory Project. And I should say that what we're trying to build is a national scale carbon inventory that can work at local scales. And so we need to put pieces together, data together, stacked between those scales. So the first thing that we do is we collect and unify existing data sets. And so, for example, we're working with Bayer Crop Sciences to gather their data, and other large ag data providers or ag services folks who have existing data sets. They're out there. And because we're a standalone science-led nonprofit, we're able to house that data without creating conflicts due to competition from for-profit enterprises. We also collect smallholder farm data. That we think that if we're going to have a view of the whole system, we need to be able to work not only at the scale of big conventional ag, but also with these small diversified holdings as well. And we think that needs to be a distributed system. It's just too hard to move people around the board. It's much easier to move bags of soil, geolocated bags, and we'll show you how that works. And then the other part of the other way that we're approaching this challenge is by combining these in a large scale modeling infrastructure to produce insights from data, turn data into knowledge. And that's where the Basso Lab comes in this partnership. The way that we do that, we have some customized field hardware, low cost, soil extractor, auger based. It's fast, gets to a fixed depth, pulls soil up, puts it in a box. And then a custom mobile application, which we started piloting with some of our field crews this summer, and that we hope to be able to distribute in the near future. We can work with partners with it starting this fall, and currently have a bunch of grants in place, hoping to be able to scale this work nationwide very soon. And this is the kind of output that we aim to give to folks and that we give to folks. So here we have sort of a picture of almost a before and after, but from compared sites in outside of Carthage, Tennessee, the home of Vice President Al Gore, his farm, which has been doing climate smart practices for about 5 years, and then a conventionally raised, very similar site, conventionally managed similar site that they just purchased a neighboring farm. So trying to look at these differences, and then a small holder in the Hudson Valley, trying to have this system work again nationwide. So talk a little bit about the campaign this summer. I want to start kind of with the idea that it takes people to get these data. And in this case, young, energetic people willing to go into weird places and weird conditions and work hard all day. And I want to particularly call out Morgan and Eliana, two of the folks pictured on the right here, who led this field campaign. But then also I was getting data and pictures back from them as late as last night, putting this stuff together. We literally just got some of the data on Friday and then the next tranche on Monday morning. And so tremendously grateful to them for being willing to turn this around so quickly. And then also just a quick call out to Scott, who I think embodies TSIP's new California summer slogan, which is going to be "Suns Out, Guns Out." So map of what we did. So we took some of these zones, thanks to Chelsea and Mark, Point Blue and Tomcat, to help us identify some treatment zones. And I want to be clear that we're not actually interested in necessarily whether the management differences in these treatments led to the effects that we're seeing, just that we knew there would be differences, because what we want to do is play with the measurements and see how they detect or don't detect these differences, at what scale, across which laboratories. And so we gathered a lot of samples. This is about 150 acres, 565 samples, obviously half that location because we did 2 depths, so 232 locations across 150 acres. This is a very high density sample. We then split those samples, homogenized them, sent them off to 3 different analytical laboratories to look at the differences that we might see between laboratories. And then based on just a morning conversation that Chelsea and I started having about some techniques that they were piloting at Point Blue, started to look at, could we get of faster bulk density using this auger. I think we have some inspiring first round numbers, but given how soon those have come in, I won't talk about them today. But I just want to talk about how we get this data. Again, field application, lay the points out, you navigate to those, you collect right in that app infrastructure, go out to the points. This is our California edition harvester, which works with a rotary hammer, which is basically a masonry drill, because it turns out that your soils are a little bit like bricks. And then we dump them into a QR coded bag. That QR coded bag helps us to track these samples from different places through the laboratory process and link back up. Again, geolocated dirt in a bag with a carbon value is a big part of our mission. And then this last part, we actually built this little dowel rod with a slider to measure exactly how deep we were actually extracting soil from, so that we could start to think a little bit about speeding up that bulk density measurement process. And the way that works, I'll just quickly play this video so that you can see what these researchers were doing out in the field. You can see they've kind of extracted that hole, and then they're filling that hole in a sock with millet. And then they're pulling that out and pouring it into a graduated cylinder to get a soil volume of what it was extracted. We can then look at the weight and the water content and try and back out bulk density. If we see that that's consistent, which it does look initially like it has been pretty consistent, particularly at the shallower depth, then we'll be able to get pretty cheap bulk density in the future. I will say that I've been in the past tremendously frustrated by California soils because they're so dense, but it looks like for this method, that's actually going to be a huge asset as compared with, say, a sandier site where it'd be harder to get predictable volumes. More on that in the future, maybe not so much today. I want to talk a little bit about the challenges that we faced, because I think this is really important when we think about fixing the soil data, the soil health tech stack. So this image on the left, that brushy area, I can tell you we got a lot fewer samples from these brushy areas. We had to dump a lot of the samples. Why? Brushy areas like that are filled with snakes, spiders, and poison oak. Nobody wants to go in there. And as we think about some of these solutions that people are talking about, oh, we're going to send a drone out. We're going to have a ranch Roomba collecting samples for us. It's going to be a robot. Picture your robot moving through there. Picture your four-wheeler moving through there. Getting into these spaces to get carbon measurements on these landscapes is tremendously difficult. How are they going to go through that fence right there, the mobile fence? How are they going to interact with those sheep or that dog? How are they going on this steep hillside where it's recently been burned? We actually found those soils were quite dry and crumbly. It was really hard to extract predictable depth out of that. These are all things that we need to iterate on. And I think embracing that iterative process as part of fixing the soil tech health stack is going to be really an important part of what we need to do. So I said that we homogenize the samples. We actually got this custom sample splitter here that evenly divides so that those could be homogenized and sent out to the 3 labs. And we knew that these 3 labs were getting exactly the same soil and see what those answers look like. So on average, we did identify, this is just Morgan and Eliana just cranked this out last night. There are differences across the treatment areas. This is the average of the 2 labs that I'm showing you here. And the reason I say average is because, big surprise, the 2 labs did not produce the same values. So it looks like there's a decent correlation between those values, but lab 1 was predictably higher across the board than lab 2. And this is something that we expected. But I think when we talk about problems to fix in the soil health tech stack, if we don't visualize the fact that labs come up with different answers given the same soil, if we don't visualize that, quantify that, and better understand the sources of that error, then we're not going to address the landscape scales challenge, right? We spend so much time talking about the variability, the spatial and temporal variability in soil carbon. You cannot address spatial and temporal variability until you can address the challenge of interlab variability. And so I think the work that we're doing here, we've got a 3rd lab we're going to have data from. This mirrors some other work that we've done at Mr. Gore's farm in Tennessee, and that we would love to continue doing it at larger scales. I think the interlab variability problem is a critical challenge for the space. So with that, I'll just wrap up and say that's what we did. And again, a huge thanks to the student and postgraduate fellows who are out on the ranch in sweltering heat all summer collecting data for this. So huge thanks to them, and thanks to all of you.
Thank you, Chris. Yeah, they did the hard work, to be honest.
They're still doing it.
Yeah, the rest of us met virtually, quite a bit, but they had to actually go out and sweat. So I'm very appreciative of the work that they've done. So, and they were really responsive last night when I had questions really late, right before this. So that thanks to them for that as well. That helped a lot. The plan was to have this all done a month ago. And as most things, that means we get it done at midnight the night before. But that was great. So thanks, Chris. Great work. That was really fun to see. If you guys have any questions, I think we're going to have a little bit of time afterward, unless Dorn is really on his soapbox today. We'll see. No pressure, Dorn, but I think we'll have a little time for questions. So if you do have some you can think of, please post them in the chat, and then we'll try to cover those towards the end. And if you know a particular person you'd like to direct it at, go ahead and direct it there. And the rest of us will try not to answer them in the chat, so that we save them for the good part at the end. All right. So with that, let's introduce Dorn Cox. Dorn, you can introduce yourself. Tell us kind of where you're going here and a look at the future, I think, and where this all fits in a big picture. So.
Yeah, thanks so much, Aaron. Just what a great pleasure to have both this conference and this topic and this great panel, which I've had the pleasure of working with in different ways. And so I think my role here as project lead for Open Team, I'll say a little bit about that, but is, like you say, Aaron, is to give some broader context and how some of these the details of sampling and protocols and everything that are put into context for some of the broader challenges that I think is part of the foundation for why we created Open Team. Just a quick check, you can all see slide deck here. Great. So I'll just quickly, I think a lot of folks are familiar with Open Team, but it's an open technology ecosystem for agricultural management project hosted and stewarded at Wolf's Neck Center for Agriculture and the Environment out of Freeport, Maine. But it's basically a recognition that the challenges that we're facing with agriculture and linking everything from the microbiome to the biosphere are large scale and require collaborative approaches, which means that they also need to be trusted and requires large scale collaboration. And so technology ecosystem is sort of a recognition that it's not individual technologies, but how they work together. And I'll coin, and you'll hear this several times, I often quote Aaron actually, is that interoperability means working together. And that means people as well as the tools and technologies. And you'll see a lot of our community within Open Team on the hackathon here and building on existing efforts and sort of embodying that approach. And then I'll just mention that ultimately, and this is sort of the bigger context, is agricultural management is the key. Is that not only is this a shared public science and intergenerational but ultimately it has to work. It actually has to work for producers on the ground and be applied. This is sort of the unique challenge that we have, is it needs to work, and not just at research farms. It has to work across all scales. So that's the focus, is actionable research. And so we're an initiative that's created with matching funding, initially launched from the Foundation for Food and Agriculture and Research, and we're now more than 45 organizations that represent diverse stakeholders that come together in the form of between 250, 300 skilled professionals whose task is to really create solutions that we couldn't otherwise tackle on our own. And so we're starting, again, recognizing that a lot of the tools and technologies that we've used to address this complex science have been fragmented across industry, across research universities, or in agricultural management. And so our task is really, again, to work together to create a coherent ecosystem. And that means not just the technology, but also, again, bridging example sites around the country. Tomcat is a wonderful example of a collaborative research site, but then linking and building on the willingness to participate in an instance like that to the kinds of data sets that Chris is beginning to aggregate, and the TSIPS project is aggregating across a number of sites. And we're working with a network of hubs, farms and ranches across the country, again, of different scales, production systems, geographies, and globally, to begin to bridge those barriers and be able to deliver the end result, which is change on the ground. And so we're really recognized, again, as we're developing technology, that we're balancing two really important things. One is that soil health is environmental data. That's really important, and that's all the stuff that you get from the lab. But to interpret it and making it useful in terms of what works where, we also have to mix that with enterprise data. Like, what were the practices that actually happened? What was the sensitivity to management? What was the management, and we'll hear more about some of this detail tomorrow, but in some of Point Blue and Chelsea's work how are we measuring differently based on historic management? That's sensitive information that's specific to an enterprise and requires a whole different data structure for us to work with. So certainly this is, and then making that both these two different types of data and their structures and how we handle them work well together is a big part of the problem solving that we've been involved with. And a lot of this work is extended back over more than a decade. But ultimately, it's how do we, again, build on this high quality in-field soil health information that's also the key to unlocking better adaptive management, improving the quality and calibrating tools like Quick Carbon or the TSIPS approaches, or sample site selection tools, improving the quality interpretation of our remote sensing data sets and machine learning data sets, and improving the quality of our agroecosystem models. All of this is part of the same, and ultimately making better decisions on the ground. So this is sort of the foundation and requires connective tissue and collaboration to make that happen. And so a lot of the work that you'll see in what we're building on, and why our team members are participating in this hackathon, is to link this high quality, this MODIS data and create a electronic data set and structure that's portable and consistent, at the farm level so that we can aggregate that data in a consistent way across these different production systems and geographies, and use that to improve the research and development, use that to increase the quality of certification programs and climate smart commodities and other attributes in the marketplace, as well as create the building blocks for environmental service claims and markets. So a few examples of some of the existing work out there that that Open Team has been involved in, and certainly a number of these that Aaron and Chris and Chelsea have also been involved in, are some of the building blocks that are part of the shared code library that the folks in the hackathon will have access to. And some of these more in this hackathon a little bit more than others, but again, is part of the shared public repository of tools to help build this coherent ecosystem to deliver these. So things like and we'll get into a little bit more of the weeds, I think, tomorrow morning. And so I'll leave some of this detail, but things like benchmarking and community platform, this farmer's coffee shop that you'll hear about, the ag data, the gateway modus standard and electronic delivery of lab results. We're currently working with USDA on the producer operational data system and creating FarmOS, the ag data management data interoperability with the USDA data collection systems to support their things like CEMAs, or their conservation evaluation and monitoring activity codes, like soil that are used to collect soil health information and aggregate it within the USDA. Sharing question libraries, and this is particularly important with and field protocols and lab protocols, so that we can create interoperability at the point of collection and a lot of the metadata that we need to actually interpret it later and be able to aggregate that. Field method protocol documentation and decision support. Chelsea was particularly involved in the development of a guide and decision support for field methods protocol documentation. And soils, and as well as some of the stratification applications that I think both Chelsea and Chris have also mentioned, so the development of improved stratification applications for sample site selection with various criteria. And then, of course, beginning to build these calibration and benchmarking libraries that we can use across the country. And so part of this on our side is also to build an architecture where we can maintain trust at the producer level and be able to opt in to be able to easily control, but also share and exchange this data from the farm level. And so that's been the foundation of our Ag Data Wallet project that allows farmers to opt in to grant conditional use of their agreement for research or for commercial purposes, or to share with peers and be able to have a full control of that system, and opt in to contribute certain portions of their data to the to public data sets to build our common knowledge and create this larger reference data for how the world works. And so some of this, again, is feeding in sort of the next stage of data visualization and management tools that certainly have a foundation in the work that the Cornell Soil Health Assessment initially did, with being able to create ratings and benchmarking your results against everybody else's. This is one of the exciting things that Modus will enable us to do across labs, to be able to do cross-lab analysis, but be able to create much more complex data visualizations that allow you to create not just everybody in the database, but be able to select who you're interested in collecting and which attributes, not just even soil health, but start looking at other aspects that are important as you're comparing and benchmarking results across the country, across production systems. And then also linking that again into a collaborative communications platform to share that data freely with control at the farmer level, and supporting anonymization. But we'll get into more of the detail in terms of that larger tech ecosystem a bit in tomorrow morning's conversation, I believe. But just wanted to give, again, that background, lots of information, lots of collaboration, lots of work over decades across agencies and research universities and ag nonprofits and environmental markets working together to have a place for the kinds of work that Aaron and his team are all doing over the next couple of days, and just couldn't be more excited about it.
Thanks, Torn. I appreciate it. And it's not just my team. There are lots of people there that I did not know before this event, which is really fun for me actually to see all these people coming together from just. It's kind of like we all just flash mob walked in off the street and started coding on soils data, which is interesting to me. But anyway, so that's been fun. Thank you, Dorn. Appreciate all your work in helping to make this event happen as well, coming up to this. So we do have a question from, well, actually, we had a question from Dennis first. There was some conversation about it in the chat about contextual data beyond the soils data. So not just the actual samples, but the stuff about it. And I just wanted to mention, we had this exact problem last night, approximately 10:30 Eastern time, that Sam and I were going through this. It's like, some of this data we have, the stuff you need to know to be able to make sense of the actual sample data has no representation in the standard for lab sample data, because that's not the point of that standard. So there's clearly some necessary piece to try to pull what experiment you were doing at the time, or what's the background behind these soil samples, and then the soil samples you have. So I have a feeling that'll be coming down the pike soon after this event. And then there was also a question from Rob I saw in the chat for Chris, asking if you could maybe talk a little bit about the struggle we had getting the soils data in time. Chris.
Yeah, I'd love to address both of those. So starting with the lab turnaround times, we sent samples to 3 labs, and we sent those, I want to say in early July, late June. We sent those samples out the door, and getting them back by, Friday and Monday involved a whole lot of emailing and calling and asking and begging and pleading and trying to move the samples forward in line. And I want to say that that's not because we chose bad laboratories. Every soil analytical laboratory that does carbon right now is completely overwhelmed with the flow. And I had a call with one of our sort of thought partners, collaborators last week, who they do soil carbon inventory, and they said, do you guys want to start an analytical lab with us? Because I mean, at the very least, sure, to control the sort of interoperable, or the interlab variation challenge that we're talking about, certainly for that part, but also we just need some control over timeline. So here you have a company that does carbon inventory talking about starting their own analytical laboratory just so that they can return maps to customers in a reasonable amount of time. I think that's going to continue to be a challenge. Every time I talk to folks who are running commercial soil laboratories, they say we're adding capacity, but we can't add it fast enough. As soon as we add it, I was saying to Rob the other day that it reminds me of the backup at the ports where the shipping containers are backed up, and somebody hears that there's capacity in San Diego, and everything from LA moves to San Diego, and then they hear, oh, there's capacity in LA, and it moves back, right? So it's really going to be a key part of the challenge. And that the second thing about the having additional layers of data, as we have this mission at TSIP, right, to try and understand how practices are linked to this metric soil carbon, and then how soil carbon is linked to things you care about, like yield and yield variability. So some of that stuff you can remotely sense. So for instance, Bruno's lab has the capacity to remotely sense yield and yield variability for the big row crops, right? So great, we can add that data on the backside. Some of the practices you can remotely sense. I can see if from space, if a field has been tilled, or if cover crops have been applied, or if it was grazed, right? But there's a whole lot of gradation in that, and there's a whole lot of understanding of what happens on the ground that you can't get remotely. And then you get into asking folks to self-report. What things are they going to self-report? What does no-till mean? What does reduced till mean? What, all of these practices, how much compost did you put down? When did you put it down? How many years have you been putting it down? What was the quality of the compost? What was, right? You can go down a rabbit hole into these additional practices. Remember that they all vary. The names vary by the place that you're talking about. The geography matters so much. And I think that is going to be one of these thorny challenges. As much as, across lab variability is going to be a challenge, I think getting these practice data into some useful format is an even bigger challenge.
Aaron,
And just how we open, some people incorrectly call pop soda. So people do that kind of stuff with soils data too. So Chelsea, I know, has some great stuff to add here as well. Chelsea, let's go ahead.
Yeah, I know there's a lot of other awesome questions coming in the chat, so I don't want to keep us here too long, but I did want to just offer that yeah, the point, Chris, that the sort of backlog in different labs and sort of how that might instigate somebody to change labs over time, I think is a really big issue, because I think from my perspective, with interlab differences for a single project, the best thing that somebody can do to ensure that your results are valid over time is to stay with the same lab. And so if a lab was once available becomes really unavailable and can't accept your samples, or says we won't be able to process them for 5 months, I think that'll become an issue that will be an issue for validity of results even within a single project if folks are moving lab to lab. So I think that's something really important. And then from a metadata standards perspective, I just want to, in my previous life, I worked a lot with molecular microbial ecologists, and I was part of the Genomic Standard Consortium. And so there's other communities who have done a lot. And so when you're working with microbial data, you have to enter it into a publicly available repository if you're going to publish. And so, and the idea there is that it's creating this public data set that then researchers and others can tap into and access and ask different questions with over time. So that's a requirement of those data. But then to your point, Aaron, and to the question in the chat, like those data are only as useful as the information that comes with them. And it comes back to ontologies and common languages and having the right metadata information there. And organizations like the Genomic Standards Consortium have created standards, and there's forms and guidance for participants who are submitting to NCBI, for instance, on how to follow those and create these interoperable data sets that can be used by others. So just to say that there could be lessons learned in other communities that we might be able to tap into there too, as we're thinking through this.
That's great. Thanks, Chelsea. That's a fantastic idea. So I believe Martha is trying to push us on to the break to start the next session. So Martha, take it away.
Thanks, Aaron. Glad to have you all here, and we'll see you over there soon. Thanks.